Hello, everyone. Good morning. Today is Tuesday, March 9, 2021, and it is 9 a.m. Eastern Standard Time. And this is the start of our program for virtual online ART training. Today, we are going to cover Module 2, Advanced Andrology Technique. My name is Ashok Agarwal. I am the director of the Andrology Center. I welcome you all to this program. We are very, very pleased to have you join us. And let me begin by my introduction. Um, I'm going to be your host for the next 2.5 hours. So let us begin. This is a brief biography about myself, and without taking too much time, I'm in charge of the Andrology Center here at the Cleveland Clinic and have been there for many years. I'm also the director of the American Center for Reproductive Medicine. I staff in the Department of Urology. We are very proud as a group. Our program is recognized as #1 in terms of the number of publications, the quality of publication, and the reach of our publications in the field of male infertility, andrology, and human-assisted reproduction. We have a very large number of medical textbooks that have been published, and we are very proud that very large number of clinicians, researchers, students have received training in our program from more than 100 countries over the last several years. I want to declare that the organizers and speakers of this program have no conflict of interest. We have not contacted any companies or outside entities for sponsorship. The program that we offer is entirely made possible by support from our center. Webcasting is supported by David Reichling and David Wolfley from the Information Technology Division of the Cleveland Clinic. Thank you to both of them. Further, the course faculty have donated their time for this online training. The program coordinators have volunteered their time for this training course. The names, images, and description of certain products or instruments shown or discussed during our presentations are being used routinely in our Andrology Center. These items are not endorsed by the speakers. This diagram or schematic shows the concept of online ART training. This entire concept came to us as we were trying to figure out what to do with the ART training, which has been part of our annual training program each year. In the month of September, we take a small number of trainees, have been doing it for the last 15 years. And many of them were writing to us, and due to the pandemic, the institution here as well as in many other parts of the United States have completely stopped making any appointments from outside for training or research related activities to protect both the trainees as well as the trainers. So we came out with this idea, and the real objective here was to offer the highest quality of educational experience in human-assisted reproduction, which is offered on a global level using all these online tools for content delivery. So this was our vision. And our goal was to raise the bar on the quality of lab skills and theoretical knowledge offered in the field of andrology and human-assisted reproduction. This really is an important part of our objective here, is how we can really do things which can improve the foundation of the services that are provided to the patients, and how we can improve the outcome of the clinics which are engaged in human assisted reproduction. And we felt that the weakest link is for those individuals who are working in the laboratory. They don't have the time, they don't have the means. A lot of training is very, very difficult to come by. They are very theoretical. They are also very expensive, and they are available only to a limited number of audience. So we looked at all these different limitations to having a model which really provides global reach. And we came out with this method. Today is our 4th online course. And we planned for 8 modules, which will be covered in 14 virtual sessions. We began about 4 or 5 months ago in November. We offered the training in November, December, and January. And today is the 4th module that we are offering today, or 4th course that we are offering. And this will continue until the November of 2022 or next year. The registration to each of these courses is completely free. The certificate of participation is issued by the American Center for Reproductive Medicine and endorsed by the College of American College of Embryology. And the faculty are drawn from our own center, as well as our colleagues and collaborators who have been working with us for many years. And the format is to provide unique type of lectures, which really are very much practically oriented, with a mix of multiple choice question at the end of the program. And then as the candidates are registered, we provide them important reading material and technical protocols. So this is essentially the type of things that we are covering. In the flow diagram you can see. The criteria to receive this special certificate of participation is a multiple choice question test. Anywhere between 50 to 65 questions will be given at the end of this course and at the end of each of the modules that we have done. And the candidates have 20 to 30 seconds to answer each question. Those candidates who take the test and score either 65% or greater will qualify to receive this special certificate of participation. You can attend the program and you don't have to take a multiple choice, but you will not receive any kind of certification. Further, there is an online survey at the end of the test, which is essentially asking the candidates to fill out an online survey. And the results of the survey in a cumulative manner are used by our program to improve the organization of the next module. So it is very valuable for us. And we are constantly updating and fine-tuning our program based on your feedback. The highlights of our last module, which was offered in January, this was on ancillary semen test. There were 6 different things that we covered: leukocytospermia, testing for sperm vitality, anti-sperm antibody, azoospermic screening, post-vasectomy screen, and retrograde sample testing. We had a fairly good number of candidates who registered, and out of that, about 735 of them took the multiple choice, and about 380 of these candidates qualified for a special certificate. And you saw those certificates being displayed with a drum beat. And I hope you can congratulate many of these candidates virtually for their fine performance. And these candidates, overall total candidates, we had 800 delegates from more than 64 countries. This program could not be run by our team alone. So we expanded our team, and we were very fortunate to have a very large number of colleagues and embryologists and andrologists who volunteered to help in this program, run this program. We have Dr. Renata Finale, who's now the chief coordinator of the program, Camila Pompeu, a embryologist from Brazil, Neha Singh, a fertility physician from India, Masha Darbandi, a reproductive scientist in Iran, Andrea Bello, she's a clinical embryologist in Brazil, Sara Darbandi, again a reproductive scientist working in Iran, Raha Sadeghi, she's here in the United States, she's studying in University of California, Sara Madani, she's again a reproductive scientist in Algeria, and Simran, who's studying again in University of California here in the United States. So these are a part of our team of coordinators who help run this program and manage dozens of things that are to be done on any given day. Their work never stops, so it will start immediately from today all the way until the next program, which will start in May of this year. So these are the highlights of our program. We began in November with Module 1A, which started with the routine semen analysis, with Module 1B, which was on sperm morphology, Module 1, which covered a variety of ancillary semen tests. And today we have Module 2, which focuses on advanced andrology techniques. This is the summary statistics. As you can see, the first bar are the number of registered candidates, and then the middle bar is the number of candidates who took the test, and the third bar in each of these is the candidate who passed the test using the multiple choice exam. So most of the candidates have done really well. For today's, we have almost 1,500 candidates now registered from over 70 countries. So we are very fortunate and very humble that so many of you from so many parts of the world, from all different continents and all different time zones are here. And I know in some parts of the world where you are coming, it may be midnight or it may be very late or close to midnight. So we thank you very much for attending this program and for your trust in our program. I just want to highlight for those who completed the training and qualified through the multiple choice, they were awarded these special certificate for their training, which is a customized certificates for each individual. A certificate is prepared which lists their name, their degrees, their institution or laboratory, and it also provides, these certificates are from the last program, it provides the course that they took, what have they learned, what were their scores that they received. So this is a pretty impressive and unique piece of certificate, which really has not been provided by anyone in the world in this particular format. So we are very proud, and I am sure that all of you are very proud to receive these special certificate, which testifies to your training, online training in a variety of courses that we are offering. You can display these certificates in your labs, in your offices, in wherever you work, and they will be part of your skills and your qualifications forever. So we congratulate all the candidates who qualified during the last program, and also we hope many of you will take this test and earn this special certificate which is really, I think, very important. Now, let's talk about what we are going to be doing today. So today we are going to be teaching and training you in advanced andrology techniques. And the techniques that we are going to cover, we are going to start by discussing what is advanced sperm function test. Then we are going to go to the most important test, which is currently being employed, is called sperm DNA fragmentation test. We'll also cover test for oxidative stress as oxidation reduction potential test. Then we are going to talk about the lab developed test for sperm DNA fragmentation, and then cover the clinical relevance of sperm DNA fragmentation in human assisted reproduction. And lastly, there will be a talk by our clinician on the clinical interpretation of everything that we are going to be covering here. So these are, again, the topics from starting in a clockwise manner: a laboratory evaluation, clinical relevance, evaluation of SDF, and lastly, clinical interpretation of laboratory tests, laboratory results that you will receive, and how to make sense of it. And this is the program in a on the left side, and our speakers for the day here. Our speakers are Professor Ralph Henkel, Dr. Sejal Gupta, and Dr. Renata, Dr. Rakesh Sharma, and Dr. Neil Parikh. So today we are going to be talking about the advanced andrology techniques. On the left side of the panel, you see the basic test, which can be covered under the semen analysis and many of the ancillary tests that you heard last in the month of January during module 1. So these are basic tests. And then we are talking about the advanced andrology test. And there are a variety of tests. This panel has just a small number of tests, which we have illustrated here. And many of them, or some of them, will be highlighted in today's talk. So why we chose the test for oxidative stress? And the reason is that there is such a large amount of information on the role of oxidative stress in male reproduction and male fertility. Right from the very beginning, from 1940 to current, there are thousands, almost close to 1,200 publications, which is probably the largest number of publications in one subject matter. So it is a very important area, and we felt that it is important for us to cover that. And also we, on the right side, you can see we have a great deal of expertise in this particular area, and we feel that we can do a very good job in talking about and explaining the practical side of these tests for male infertility. And then the sperm DNA fragmentation, another important test. So if you look at the left side, again, this is a very hot area with very large number of publications, both original publication reviews and meta-analysis. And this data is only until 2019, and certainly the number have increased since then. And on the right side, again, we have at the Cleveland Clinic a great deal of expertise in this area, not only from a research point of view, but also from a clinical point of view. We have developed techniques, we have standardized those techniques for sperm DNA fragmentation testing, and we felt that we can share these things with a great deal of expertise and confidence. Lastly, you can connect with our center by sending a request, searching my name and Cleveland Clinic. And the benefit of connecting is that we are providing daily updates on our research activities. We provide free access to hundreds of research articles published by our center, by our scientists and researchers and our collaborators. You can also read the latest research which have been presented at important national and international meetings, reproductive meetings, and urological meetings. Learn about virtual research opportunities that our center provides. Learn about our online ART training courses, and also the opportunities to collaborate with a team of ACRM researchers. So please consider joining on our LinkedIn. It is my great pleasure and honor to introduce our first speaker, Professor Ralph Henkel. Dr. Henkel is currently a visiting reader in the Department of Metabolism, Digestion and Reproduction at the Imperial College of London, United Kingdom. He also serves as the Chief Scientific Advisor at Logix Pharma in Reading, United Kingdom. He is the extraordinary professor of reproductive biology in the Department of Medical Biosciences at the University of Western Cape, where he served in that capacity as a full professor for almost 20 years. He holds a number of other positions. More importantly, he is the editor-in-chief of Andrology, one of the most prestigious original andrology-focused journal in the world. He is well known all over the world in the field of reproductive medicine and infertility with very large number of publications. And he has a great deal of experience in teaching and training, and he has been working in with our program as a scientific collaborator and a research collaborator for a large number of years. So it is with great respect, and I would like to welcome Dr. Henkel. He has recorded his presentation, although he is live connected with this program, but he has recorded his presentation to avoid any problem with the internet connection speed. So we are going to play his presentation.
Good morning, good afternoon, good evening, everybody. Since my presentation on advanced sperm function tests is an introduction to these tests, following lectures will go into more detail of these specific tests. Coming to the first part, that is the learning objectives, the limitation of the semen analysis, and therefore there is a need for new sperm function tests, which include an advanced test that is part of oxidative stress testing with mitochondrial membrane potential, reactive oxidant species, ROS, total antioxidant capacity, lipid peroxidation, and oxidation reduction potential, or ORP. On the other hand, we are talking also about sperm DNA fragmentation, which includes the Agglutinin orange test, the SCSA or sperm chromatin structure assay, the SCD, and the tunnel and the comment assay. And last but not least, proteomics and bioinformatic tools, which are new developments. Since there is a definite need for new advanced tests, this is because the standard semen analysis provides only very limited information about the sperm fertilizing ability. And this is what we see mainly with normal semen analysis. They may be fertile or infertile. So what we see here is shown in this figure here at the bottom. There's a huge overlap between patients with normal sperm count or normal parameters and those that are normal fertilizers. So we cannot clearly distinguish between patients with good fertilizing ability and those who will have a problem. Therefore, the normal semen analysis has limited predictive value for the pregnancy outcome. And therefore, actually, we need more tests that provide relevant information on sperm functionality. All these tests here on the left-hand side, they are coupled with a standard semen analysis, which is concentration, morphology, motility, and so on. But these tests do not give information about the functionality of a sperm. So that means there is no information about sperm DNA fragmentation, reactive oxidant species, mitochondrial functionality, sperm zonal binding, chromatin condensation, acrosome reaction, or olemma binding. Therefore, these sperm functions must be tested for in specific individual tests. And now, at this stage, we also need to distinguish between the area before ICSI and after ICSI. Because before ICSI, in the pre-ICSI area, all these tests had to be done individually, like acrosome reaction, zonal binding, sperm vitality, eosine nitrogen staining, sperm olemma binding with a hamster penetration assay, lipid oxidation, or even the acrosome test to test for the sperm ability to penetrate the zona pellucida. After the introduction of ICSI, many of these tests fell away because we just bypass the zona pellucida. So there's new tests that were developed, like Ros or ORP, the oxidation reduction potential, or total antioxidant capacity and total antioxidants, sperm DNA fragmentation, hyaluronic binding, fluorescence in situ hybridization, and with a future perspective, the proteomics. But let's start with the mitochondrial membrane potential. Mitochondria are the powerhouses of any cell, as for sperm cells, so they produce all the energy, and therefore they need to be functional. And this functionality of the mitochondria can be tested with a fluorescent dye, which is called JC1. In normally functional, meaning healthy mitochondria, this JC1 aggregates to larger complexes which fluorescence in a fluorescence light red. If this aggregation cannot be done due to mitochondrial problems and dysfunctional mitochondrials, then the fluorescence is green. So we can actually quite nicely distinguish with this JC1 between healthy cells with a healthy mitochondria, which then appear with an intense red fluorescence, and unhealthy cells with unhealthy and damaged mitochondria and mitochondrial membrane potential, which then fluorescence green. And this can be shown in a direct linear relationship with the cell's own Ros production, that is negative correlation. The higher the mitochondrial membrane potential, the lower the Ros production, the more healthy is the cell. So healthy sperm cells from patients we will rather find in this area. From donors we find in this area, patient sperm we will find in that area. But I mentioned reactive oxidant species. What are reactive oxidant species? That are highly reactive or extremely reactive molecules with an unpaired electron in the outer orbit. So here this displays just a normal stable molecule, and if it loses one electron here, it gets one unpaired electron, which makes it a so-called free radical. And this radical status makes it highly reactive, reacting with lipids, proteins, and nucleic acids, so with the DNA. And this is what we can then see in the semen or in other cells. in the semen, these reactive oxidant species are produced by abnormal spermatozoa and by leukocytes or white blood cells. Here on the right-hand side, I'm showing you some examples of reactive oxidant species and some other derivatives thereof, like reactive nitrogen species. If these reactive oxidant species are now available in too high concentrations and not counterbalanced by antioxidants, leaving the system in an imbalance between reactive oxidant species and the antioxidants, this is then causing oxidative stress. These ROS can come from endogenous sources like the spermatozoa or immature spermatozoa and leukocytes, or exogenous sources like smoking radiation or toxins. So when we see oxidative stress, that is then pushing the bodily balance of this balance here into an oxidative stress situation with an overwhelming amount of reactive oxidant species, that is then causing damages to blood membranes in form of lipid oxidation, damages to the DNA, or damages to proteins, leading eventually to this sperm dysfunction and infertility. But how can we measure reactive oxidant species? There are different techniques available. The most common technique is luminometry. And you can see here a luminometer. We use the luminol as a luminescence probe. This technique is then called chemiluminescence. And with this technique, we can clearly distinguish between fertile donors and infertile patients. The higher the Ros production, the more then it's infertile patients, and the cutoff value is here by less than 102 relative units per second per million sperm, but for normal Ros production. And with this cutoff value, we can clearly distinguish between proven fertile donors and patients, and here even proven fertile donors and patients with oligozoospermia. And there is a negative correlation between these variables. The cutoff can be used as a diagnostic or screening tool to diagnose male factor infertility. And that will then give the means to actually treat the patients with antioxidants if the level of reactive oxidant species is too high. But on the other hand, I mentioned that this oxidative stress is a balance, and the body needs to be in balance. It's not only the reactive oxidant species, it's also the antioxidants. So oxidative stress can also be caused by a lack of antioxidants. And we can measure these antioxidants with different tests. The test is the total antioxidant capacity. It's a calorimetric assay with using Trolox as a reference, and the normal range should then be more than 1,900 micromoles of this Trolox. And with this, we can also clearly distinguish between fertile donors that have a relatively high amount of total antioxidant capacity versus infertile patients where this total antioxidant capacity is significantly reduced. And there are also different other techniques available to measure damages. In this time, the measure and the damages to the plasma membrane in terms of lipid peroxidation. Here we have a test for barbituric acid assay, or TBOS, measuring isoprostane, or here HNE his adducts But all these tests here, although they have some advantages, they have significant disadvantages. They are labor intensive, they're expensive, and they are not really clinically validated. Another method that basically measures both sides of the coin, which is the oxidative stress, as well as an excessive amount of antioxidants, which would then amount to reductive stress, which is the ORP, or oxidation reduction potential, which is measured with this Myosis system, with this device here, and these sensors. The principle behind this technique is that the device is measuring the transfer of electrons from reductants, which are the antioxidants, to the oxidants, or the ROS, and the result is then displayed in millivolts here on this display. And then we just need to normalize this result with a sperm count in millions per milliliter, and the cutoff value that was calculated is less than 1.34 millivolt per million sperm per milliliter. If we have now high oxidative stress levels, that is causing DNA damage, and there's different types of DNA damage available, but this can always come from different sources. There's intrinsic factors like defective maturation, cancer, diabetes mellitus, infections or varicocele, or extrinsic factors, which is poor diet, poor lifestyle conditions, smoking, alcohol, etc. All these conditions are causing then high levels of reactive oxygen species, which is then causing DNA damage. And there, like I said, there are different types of DNA damage, like single strand breaks, damaged bases, mismatch, intra-strand crosslink, double strand breaks, inter-strand crosslinks, and so on. And all these together are then causing major problems with the assisted reproduction outcome. The most discussed DNA damage is DNA fragmentation, for which I want to briefly introduce a couple of tests, which is number one, and the most commonly used test is the tunnel assay. Then there's acodine orange test, or the sperm connoting structure assay, or the SCSA. Then we have the sperm connoting dispersion assay, or SCD, and last but not least, the COMET assay. And all these tests, they're measuring single and double strand breaks, and they can either be detected with a microscope, or with a flow cytometer, or just on a normal microscopic slide. So here is the tunnel assay, most commonly used with the flow cytometer, the same as the SCSA. Here the SCD test is a slide-based test, as is the COMET assay. Coming to the tunnel assay, the tunnel stands for terminal deoxynucleotide transferase DUTP NIC end labeling. So this test uses a fluorescence dye that binds the DUTP to the open strand breaks, that open nicks, labels it, and can then be identified in the either fluorescence microscope or in the flow cytometer. And there we can also clearly identify fertile group versus infertile patients. The cutoff value here is less than 17% of DFI, or DNA fragmentation index. Coming to the next test, which is the acodine orange test, the principle here is based on the metachromatic characteristics of the acodine orange. which binds to the DNA, and if this DNA is normal, it fluoresces green, like here, these sperm. If it is damaged, then it fluoresces orange to red, and thereby we can distinguish between these different sperm groups. The test is quick, it's simple and inexpensive. On the other hand, there are also inter-laboratory variations and a lack of reproducibility. The most standardized test is the sperm chromatin structure assay, or SCSA. It is the same principle, but it's using the like the aqualine orange, but it's using the susceptibility of the sperm DNA to denaturation. So it's not determining real DNA damage, it determines potential DNA damage. And here the cutoff value is less than 25% of red stained sperm. And here we can clearly differentiate between patients that are not pregnant. Here we can see these high amount of sperm which have red fluorescence, whereas here it's pregnant patients that is less of these and more of the green sperm. There's a strong correlation with other sperm DNA fragmentation tests, and on the other hand, it's inter-observer and inter-laboratory variability was not tested, and it's technically a bit demanding. An easier test is the sperm chromatin dispersion test, which is a slide-based test where we have agar rows, embedded sperm, and normal DNA is then under these denaturating conditions, forming loops, which in the end can be seen as these halos around the sperm head, and can then either be stained or just directly measured in the microscope. So if there is no halo formation, then that's an indication that the DNA is damaged. If there's a large halo like this sperm here, that is good DNA. Advantage is standardized technique. It's quick. It's established clinical threshold about 24.7 or 25%. It can be performed on fresh and frozen samples, but on the other hand, it has disadvantages like indirect assay involving acid denaturation. Is also not determining real DNA damage, it's determining potential DNA damage. And there's also inter-observer variability. Coming to the COMET assay, which is a more advanced and more complicated assay because it uses electrophoretic assessment. We need an electrophoresis chamber. It can either be done in an alkaline or neutral conditions. And the name of this test comes according to what we see in the slide. The more the DNA is damaged, the more fragments we will see, and these smaller fragments, they move out of the sperm head, and in the electric field, they then form this comet-like structure, which actually gave the test the name. A more complicated test is when the combination of the SCD with fish, where we can then not only see general DNA damage, we can also clearly identify with the fluorescence probe for the fish test where the damage is situated, if there's perhaps a chromosome damage or a specific gene damage that can then be identified here as these fluorescent signals. Another test that is already selecting spermatozoa in terms of the sperm selection is the hyaluronic acid test. If we look at the cumulus ovaris, that is the cell mass that is surrounding the oocytes, this cell mass contains a lot of hyaluronine, and sperm bind to hyaluronine. And only normal sperm could bind to this. So therefore, the test was developed in a petri dish where the petri dish is covered with hyaluronine in a chamber, and sperm bound to this hyaluronine. They are highly motile. They are vigorously beating with the flagella. They are not acrosome reacted, and they show really here good DNA integrity. So this DNA is not damaged, as can be seen here in this acridine orange stain. Coming to more futuristic techniques which have started being evaluated recently, which is in the omics age, where we look at proteomics and the protein expression and protein damages, and then to differentially expressed proteins. And we can clearly see here the green bars are indicating patients with low ROS, whereas the red and even then the blue bars indicating patients with high ROS activity in the semen, and this causes a change in the proteomic expression. So we can see here clear differences between these patients with low ROS and low oxidative stress as compared to those patients with high oxidative stress. And then these proteins can be analyzed with specific proteomics techniques, and then we can see here clearly what can, if there's an overlap or if there's a shift in specific proteins. Last but not least, for all these techniques that are used, we need to look at the quality control, not only for all the instruments, for luminometer, for ELISA reader, for flow cytometer, Myosis Analyzer, or the electrophoresis. We also need to check for the quality of the reagents that we are using. And of course, the staff in the laboratory must also be competent and must be regularly assessed, and the competence must be shown and recorded. A look into the future, I mentioned it already, the proteomics look very promising, but there are also other omics techniques available which are currently under investigation, like transcriptomics, telomere length evaluation, genomics, epigenomics, DNA protemination, or possibly single nucleotide polymorphism genotyping. So that is possible development, but I'm sure that will still take a couple of years until we have some of promising and validated techniques in andrology. And with this, I come to the take-home message, which is a basic semen analysis is a poor predictor of fertility. Therefore, advanced tests have to provide additional information on sperm fertilizing ability. There's an imbalance between reactive oxidant species and antioxidants, which results in oxidative stress. And if there's oxidative stress, then this will lead to damaging of the plasma membranes, of lipids, of the proteins, and the DNA. These can then be tested with the SCSA, COMET, SCD, and tunnel assays as markers of DNA fragmentation. Proteomic tools can be used, but are still under development. And finally, the use of advanced function tests may then help identify oxidative stress-related causes of infertility. And with this, I thank you very much for your attention.
And we move on to the next, but before that, I want to tell you that we are in my office here orchestrating this program, and the next speaker will come under my name, but and I moved to another chair. So if you see him, he is my associate here. His name is Dr. Rakesh Sharma. Rakesh is an associate professor at the Lerner College of Medicine of Case Western Reserve University. He is the coordinator and assistant technical supervisor of our Andrology Center. He has published a very large number of articles and research before moving into the clinical area completely. And he has received several awards and is well recognized in the field of andrology. So Dr. Sharma or Rakesh is going to be, please next slide, is going to be talking about the laboratory evaluation of oxidative stress. So I give this now for Dr. Sharma to take it and start his presentation.
Thank you, Dr. Agarwal. Good morning, everyone. The talk will be on laboratory evaluation of oxidative stress. The learning objectives are to understand oxidative stress and its role in male infertility, examine some of the direct and indirect tests of oxidative stress, review the current challenges with the current markers in measuring oxidative stress, introduce a new marker of oxidative stress called the oxidation reduction potential, or ORP. I will also highlight some of the lab scenarios that we encounter when we use this test. I will also review the quality control associated with this test and highlight some of the advantages and disadvantages of ORP. Male infertility can be attributed to a number of factors. They could be the lifestyle, alterations, genetic changes, hormonal causes, varicocele, systemic diseases, but in 50% of the cases, the underlying etiology is unknown and they present with abnormal semen parameters. In these patients, oxidative stress has been shown to be a major player. Oxidative stress can be caused because of a number of factors such as pollution, drugs, smoking, altered semen parameters, infections, presence of varicoceles, cancer, and they all lead to oxidative stress because of the presence of free radicals such as superoxide anion, hydrogen peroxide, hydroxyl radicals, and they cause an imbalance resulting in oxidative stress. And this oxidative stress causes the alterations in the proteins, the lipid peroxidation, apoptosis, DNA fragmentation, resulting in sperm damage, and eventually resulting in male infertility. In this slide, I would like to explain to you the mechanism of oxidative stress associated with male infertility. The white blood cells are the main contributors of reactive oxygen species. They result in inflammation or infection, resulting in the release of cytokines and the generation of reactive oxygen species. The other source is the production of abnormal sperm during spermatogenesis, which contain the cytoplasmic droplet. And this cytoplasmic droplet results in altered glucose 6-phosphate dehydrogenase, resulting in increased production of reactive oxygen species. Also, the apoptosis going on during spermatogenesis triggers the caspase activation process, resulting in abortive apoptosis, and they all contribute to the DNA damage, damage to the proteins and the lipids, altered sperm functions, eventually resulting in male infertility. Oxidative stress can be measured both by direct methods such as chemiluminescence, nitroblue tetrazolium, cytochrome C, oxidation-induced fluorochrome probes, and also it can be measured by indirect tests such as the myeloperoxidase test, also known as the ENDS test, measuring the lipid peroxidation levels, measuring the chemokines, measuring the antioxidants as well as the total antioxidants, and also measuring the DNA fragmentation using these four commonly used methods. Each test has its own advantages and disadvantages. And for example, the reactive oxygen species measurement by chemiluminescence, it is a robust, highly sensitive and specific test. And luminol has the ability to measure both the global ROS levels, but it also has the disadvantages that it is time consuming, requires large and expensive equipment. And similarly, the other tests also have their own advantages and disadvantages. In this slide, I would like to show you what are the commonly used current markers. And these are the reactive oxygen species, the total antioxidants, the lipid peroxidation method, apoptotic markers, DNA fragmentation, as well as protein oxidation. However, all these markers have inherent challenges. And these challenges could be that these are individual markers. They capture only one dimension of oxidative stress. They do not provide a complete picture of oxidative stress. They reflect redox status at a given point. They're time consuming. Many of them are outdated, time sensitive, expensive, and they require special training. So we are looking for an ideal marker of oxidative stress. And what are the attributes? Some of the attributes that we're looking for is that it should be a single marker. It should provide us the real-time redox status. It should be simple to use. It should be a rapid test and allow us to measure the oxidative stress in both fresh as well as in frozen samples, and should have the ability to measure and serve as a surrogate marker of semen parameters. And one such test is the oxidation reduction potential, or the ORP test, which is used by using the male infertility oxidative stress system analyzer. And the principle is based that it can measure the overall balance between the total oxidants and the total reductants. It's a galvanostatic technology. And once the sample is loaded, it eventually reaches the reference electrode and the circuit is completed and a low voltage current is generated. This is the Myosis Analyzer, and this is the sensor with the sample port where the sample is loaded and it reaches the reference cell, which then completes the circuit, and we get the display of the voltage, which is a reflection of the level of oxidative stress. Here I would like to show you a quick video clip. This is the Myosis Analyzer, and this is the sensor. We remove the sensor, we mix the sample, and we place 30 microliters on the loading chamber. And this sample then moves to the reference electrode, completing the circuit, and we will get the read for the ORP in about 4 minutes. So this gives us the reading of the ORP once the sample is placed in the sensor. As Professor Henkel mentioned during his lecture, that ORP by itself is not very meaningful, and it is important to normalize the ORP, and this is accomplished by dividing the sperm concentration with the ORP, and this gives us the adjusted ORP, and the reference value of the adjusted ORP less than 1.34 millivolts per million sperm is the reference value, and any values higher than this, they indicate the presence of oxidative stress in that given sample. We encounter different situations when we measure the ORP in any given setting, and today I would like to illustrate a few of these situations by giving you some scenarios. So scenario number one is that a semen sample has a volume of 1.5 ml and there is no sperm present in the wet prep. What will you do? The solution to this problem is that the ORP cannot be normalized because there are no sperm seen in the ejaculate, and therefore we make a comment in the patient's report that the ORP cannot be measured because of lack of sperm in the sample. Scenario number 2 is that the sample is highly viscous, the semen volume is 1.2 ml, and the analyzer displays error messages. What will you do? Highly viscous samples, they cannot complete the circuit between the sample and the reference port, and therefore the electrode connection is not made and we do not get any reading. So again, we make a note in the patient report that the ORP cannot be measured. And the other scenario is that a semen sample has a volume of 2.5 ml. The concentration is 110 million per ml. What will you do? Now, in this situation, it is easy that we simply dilute the sample and we factor in the dilution factor into the normalization calculation, and that will provide us the ORP value in that sample. Scenario number 4 is that the semen sample has a volume of 1.5 ml, concentration of 40 million per ml, and the analyzer gives an error message. What we will do in this situation? Well, the solution is that the circuit has not been completed. So we simply remove the sensor and reinsert it gently, and the circuit will be completed and we will get the ORP reading. Scenario number 5 is that semen sample has a volume of 1.5 ml. It is severely oligozoospermic with a concentration of 5 million. What will you do? Well, we measure the absolute ORP and we normalize it by dividing with the sperm concentration. In this situation, the ORP will be very high, indicating the presence of oxidative stress. Scenario number 6 is that semen volume has a volume of 1.5 ml, concentration of 40 million per ml, and the presence of white blood cells of 6 million per ml. What will you do in this situation? The solution is that we measure the ORP and we divide it by the sperm concentration. N's test or the presence of white blood cell is not included in the calculation, and therefore no further adjustment is necessary in this case. Quality control is very important in the measurement of this test, and this can be accomplished by making use of the Calibration Verification Key. This is the Calibration Verification Key, which simulates the presence of the actual sensor. It has two sides, side A and side B, which have different voltages, and side A has the range of 99 to 101 millivolts, and side B has the acceptance range of 295.8 to 304.2 millivolts. So when we insert the sensor with side A or side B, if the reading is in this range, then the quality control passes. This sheet provides the daily or the ORP when the test is done, and each time we measure the ORP for side A and side B, and they should fall in this range, and the technologist will then initial indicating that the quality control has passed. Another way to perform the quality control is the use of the low and high solution ORP. The low solution has a voltage of 33 to 70 millivolts, whereas the high solution has the voltage of 91 to 117 millivolts. So we place 30 microliters of each solution on the sensor and we read the voltage. The quality control sheet will have the lot number, the date that the sample quality control was performed, the high solution ORP and the range, and similarly the low solution range. And if it passes, the technologist performing the quality control will initial indicating that the ORP has passed the quality control. So the advantages of the ORP test are: it's a real-time measure of redox status. It is simple, rapid. It can be measured in fresh and frozen sample, as well as in seminal plasma. It also serves as a surrogate measure and measures all antioxidants present in a given sample. This slide shows the distribution of normal and abnormal semen parameters in patients and the cutoff of 1.34 that is used as a reference value. And you can clearly see that all normal samples are below the cutoff of 1.3 millivolts, whereas patient samples with abnormal semen parameters show higher ORP levels. In this slide, I would like to draw your attention to a study that was performed in our center with a sample size of 807 subjects, in which the samples were distributed into two groups. Group 1 was those with normal semen parameters, and Group 2 were the patients with abnormal semen parameters. And as you can see from here, the ORP in the first group with normal semen parameter was very low, which is 1.34, and all patient group with abnormal semen parameters had elevated levels of ORP, which were very significant. In another study done by the Hamad Medical Corporation, a sample size of 4,000 subjects were included, and these were divided into two groups. Group 1 were those with normal zoospermic samples, and the other group were those with the non-normal zoospermic samples. And as you can see from here, the ORP levels were significantly higher and almost 5-fold higher than those seen for the normal zoospermic group, indicating that ORP is a good indicator to measure the semen parameters in the two groups. In another study by the same group, the samples were divided based on the sperm motility into normal sperm motility group and those with poor sperm motility or the astenospermic group. And again, the distribution of ORP was significantly higher and 5.63 millivolts compared to the 1.79 millivolts in the normal sperm motility group, again indicating that the ORP measure is different in the two groups of patients shown over here. In this slide, I would like to show the importance of oxidative stress measure using the ORP measure as a test for oxidative stress. Almost 46 million infertile men have some underlying cause of oxidative stress, which can be measured using the reactive oxygen species test. But there is another group, which is the idiopathic group, comprising of almost 46.5 million patients. And these are the patients that have abnormal semen parameters with no known cause of infertility. In these patients, oxidative stress was found to be the underlying factor in 80% of the cases, which amounts to almost 37.2 million patients. And these patients would have not been diagnosed if we did not measure the oxidative stress using the ORP as a measure. Therefore, we categorize this group of idiopathic infertility as the male oxidative stress infertility, or a new group called the MOSI, and this gives the worldwide incidence of MOSI, which is a categorization of patients who otherwise are termed as idiopathic. So using the ORP as a test, these patients are actually the patients who will demonstrate high levels of oxidative stress, which then can be treated with antioxidants and managed. So to leave you with some take-home message, oxidative stress is the underlying cause of male infertility. Current markers of oxidative stress are unsuitable to be used as a diagnostic test, and ORP can distinguish both the infertile patients with normal and abnormal semen parameters, thereby serving as a surrogate marker of semen quality, and high ORP is the indicator of MOSI, which then be treated using the antioxidants. Thank you very much for your attention, And this is another view of the Cleveland Clinic.
Dr. Gupta is, as a brief introduction, is the technical supervisor of our Andrology Center and Reproductive Tissue Bank since many years. She has a number of fellowships in reproductive medicine, which was from WHO and then followed by a research fellowship. She has earned numerous awards, as you can see here. And she also has done her master's in embryology from the Jones Institute. She has published a large number of articles. And today she is going to speak on, let's have the next slide. So her presentation today is going to be about the clinical relevance of oxidative stress in male infertility. And she's going to build upon the presentation that Dr. Sharma gave, which covered the laboratory aspects. She's going to talk and relate this laboratory findings into what is the clinical meaning of oxidative stress in male infertility. So, Dr. Gupta, Sejal, you can take it from here.
Well, thank you so much, Dr. Agarwal. And the topic has been described by Dr. Agarwal. So basically looking at clinical impact of oxidative stress in infertility. The learning objectives of my presentation are seminal oxidative stress impacts semen quality and leads to infertility. So the first objective is to learn about the use of oxidative stress markers, both as a diagnostic and prognostic tool in male infertility. Secondly, look at oxidative stress and what is the impact on natural conception or natural fertility. And thirdly, look at varicocele, which is an important condition seen in infertile males, and what is the role of oxidative stress and sperm DNA fragmentation in this condition. Oxidative stress generation is known to occur in the ART conditions, and hence examine what is the clinical impact of oxidative stress on male infertility and ART. And what are the ways and means that we can control or modulate oxidative stress in the ART lab and have optimal gamete quality as well as clinical outcomes. So here, this is an illustration which shows that physiological role of reactive oxygen species. They influence sperm function, they have an influence on sperm maturation, cause sperm motility, capacitation, acrosome reaction, as well as fertilization. So important physiological impact of normal levels of ROS. On the bottom, you see the unregulated ROS production. And this can cause oxidative stress when the balance is tipped and there is increased ROS and the antioxidants are not able to neutralize. And this results in lipid peroxidation, DNA damage, and apoptosis. Now, if we examine the causes of increased generation, exogenous causes are radiation, toxins, smoking, and alcohol. So a lot of lifestyle factors, whereas the endogenous sources are leukocytes, and these lead to increased NADPH levels, which in turn lead to increased ROS levels. Immature spermatozoa, through the excessive residual cytoplasm, can generate ROS and varicocele, which causes testicular hyperthermia as well as hypoxia and leads to reactive oxygen species generation. So here I want to present a study which examined and investigated the correlation of two markers, the ROS and ORP, and whether these were commensurate in their assessment of oxidative stress in male infertile patients as well as normal subjects. And as you can appreciate on the right, there is this bar plot which shows that there was significantly higher levels of reactive oxygen species in men with abnormal sperm parameters. And the highest level of ROS were seen in leukocytospermia, which was assessed by a positive ENS. And there was significant positive correlation between observations for ROS and the ORP levels, as seen by the p-value shown here. So further, this is the second box plot, which shows the median values of ORP levels and the interquartile ranges, again showing that men with abnormal semen parameters had significantly higher levels of ORP compared with the normal sperm parameter group. So this is a second study that I want to present here, looking at investigating the oxidative stress markers and the semen parameters. What is the relationship? The oxidative stress marker examined here was the 8-hydroxydeoxyguanosine, and there were men who were normal subjects, as well as different abnormalities of the sperm parameters, which were categorized as shown here. So what this study showed was basically that the 8-hydroxydeoxyguanosine levels were significantly higher in men with abnormal semen parameters, be it as theozoospermia or oligozoospermia, compared with the normozoospermic men. And overall, the study showed that there were higher median values of 8-hydroxydeoxyguanosine in the samples which had abnormal semen parameters compared with the men who had normal semen parameters. And also the study, the significant finding was there was a negative correlation between the 8-hydroxy, so an inverse relationship between 8-hydroxydeoxyguanosine and the sperm concentration in this group of patients. So varicocele is an important condition in general population, seen in about 15% of men. In infertile men, it is higher, about 35 to 40%. So this study was from our group, and we looked at the marker, the ROS levels in male infertile patients compared with the controls, which were healthy men. And what were the significant findings is that there were higher ROS levels that were seen in all patient groups compared with the controls. But the highest levels of ROS were reported in the, if you focus your attention on the graph on the right side, the highest levels were seen in the varicocele associated infection group, and this was highly significant. The last finding was that there were high ROS levels also seen in men with oligozoospermic samples. So another study which examined the role of oxidative stress in varicocele was looking at varicocele patients within two groups, normal semen parameters versus abnormal semen parameters. And the ROS levels were high in both groups of varicocele patients, one with normal semen parameters and abnormal semen parameters compared with the controls. Looking at the second image, which is at the bottom, if you see, they also looked at the percent DFI levels by the SCSA and the tunnel test. And both showed that the percent DFI and the percent tunnel positivity was significantly higher in the varicocele group, whether they had normal semen parameters or abnormal semen parameters. And so here, going further, this study examined what was the effect or impact of seminal ROS levels on natural pregnancy. So this, the marker was, the clinical outcome was natural pregnancy, and this group of infertile patients were followed up for 24 months. And what this study reported was that the ROS levels were significantly higher in the non-pregnant group compared with the pregnant group. On the right side is an illustration which shows, and the study investigated the comparison of pregnancy rates between the two groups that were above the cutoff level, which is high ROS levels, and below the cutoff level, so they had low ROS levels. And the blue is the number of pregnancies that was seen in the group with the low ROS levels, which was significantly higher than patients who had high ROS levels. And this was integrated measurement of the ROS for over 30 minutes, and the cutoff, as shown here, was 4.35 millivolts per million sperm. So this presentation, I want to also emphasize that ROS is generated in ART conditions, in the culture conditions, and there are several factors which can lead to ROS generation. So if you examine over here and focus your attention, any change in the pH and temperature, if there are shifts, the oxygen concentration, as well as the culture media, can lead to generation of ROS and impact the gametes and the developing embryo. And within the culture dish, the embryo itself, because of certain toxins that are generated, or even the culture dish itself leads to generation of certain organic compounds, which can impact and lead to increased ROS generation. Centrifugation of the sperm sample for preparation also is a known cause of increased reactive oxygen species. And all of these can impact the spermatozoa in terms of causing sperm dysfunction. Oocyte quality can be impacted, and the embryonic development may get blocked, or the embryo can undergo apoptosis. And on the right side is this impact highlighted, that oxidative stress with high supraphysiological levels of ROS can impact both the spermatozoa, the oocytes, and the embryos, with decreased cleavage and apoptosis that can result in the embryonic development. So this was a study which was conducted by our group, and it was done to compare the oxidative reductive potential in different ART culture media. So we looked at 2 of the sperm wash media, as well as other media that are used for oocyte culture, as well as for embryonic culture. So if you shift your attention to the graph, which is on the right side, what one can appreciate here is that the SAGE 1-step medium, which is used for embryonic culture, had the lowest ORP levels, significantly lower than if you look at these 2, which are the sperm washing media from different vendors that had the highest levels of ROS. So taking this process forward, our group with a collaborator also then went on to examine what are the ORP levels in the follicular fluid and the culture media that is used for oocyte culture after collection. So the study design was to examine the follicular fluid, and this was examined both in oocyte donors as well as infertile patients, and IVF media, which was a wash, and a oocyte culture media. So looking at the graph on the right side, it's shown that the follicular media had 86 millivolts of ORP levels, and these were much lower than the global total media, and this was significant. So this helps us to think that maybe the oxidative stress levels within the culture media need to be modulated to lower levels. And further, the study also found that infertile patients had much higher ORP levels compared with the oocyte donors. And within the different 2 media that were examined, the culture media had lower ORP levels compared with the wash media. So the conclusion of the study was that follicular fluid ORP levels are significantly higher in infertile patients versus oocyte donors, and some amount of modulation of the ORP in the culture is needed to adjust and increase the success rates of IVF. So taking this further, our group with a collaborator also designed a study. The goal and the objective of the study was to look at the impact of supraphysiological levels of ORP in human embryonic culture media and how it affects the blastocyst development in IVF. So the design of the study was to 104 patients were recruited. These were women less than 37 years who were undergoing IVF cycles. And they were, this was a prospective cohort study. And there were two groups, each had 52 patients. So in the one group, which is at the top, the embryonic culture media, the ORP was adjusted with the addition of certain cocktail of antioxidants. And the outcome looked at was blastocyst development rate, whereas the bottom is the group where there was no adjustment of the embryonic culture media. So let's look at the results. So here are the results from this study conducted for 104 patients. And the first figure shows the blastocyst formation of embryos that were cultured. And these are the embryos that were cultured in the media with the adjusted ORP versus media not adjusted. So there's a BDR rate was significantly higher in the ones which had the adjusted ORP in the culture media. The second clinical outcome that was examined was the ongoing pregnancy rate from embryos that were cultured in adjusted versus the non-adjusted. Again, significantly higher where there was an adjustment done. So how was this adjustment achieved? The figure on the right, you can appreciate the blue line shows the ORP without the antioxidants. So this was a cumulative measurement of ORP in the culture and the culture media for the blastocyst. And the green is where the ORP adjustment was done with every 12 hours, there was supplementation of the antioxidants. So this is how these results were achieved, because just single addition of the antioxidant did not have a persistent ORP level, which was kept around 90 millivolts. So the goal was to keep the ORP around 90 millivolts, and this was achieved by adding the antioxidants every 12 hours and achieving the successful outcomes. So further, effects of seminal ROS have been examined by a study on embryonic development after ICSI. And the seminal ROS levels were significantly lower. If you look at the box plot on the right side, in the poor quality embryos, the levels were significantly higher compared with the good quality blastocyst group. And the second important finding of the study was that the cutoff levels of ROS for good cleavage embryo were 6,601 versus good quality blastocyst 2. So achieving a good quality blastocyst requires even lower levels of ROS in the semen sample, and that was one of the conclusions of the study. Lastly, I would like to present this study, which is from 50 patients who underwent ICSI, and a receiver operating characteristic curve was generated for the ORP levels measured in the semen sample. So here, this is the ROC analysis that was done, and the findings were the cutoff that was generated from this ROC analysis was a cutoff of 0.37 for the ORP levels, and the area under the curve is pretty predictive. It is 0.6. It's not the best, but it does have some predictive value for predicting pregnancy in this group of patients. I just want to highlight that this cutoff is much lower than the 1.34, which is used in the semen samples, because within the ART settings, there are multiple factors that can affect the embryonic development and pregnancy rates. And these include the oocyte quality, as well as the ART culture conditions, and of course, the seminal ROS levels. So overall, the take-home message from this presentation is, as shown by several of the studies, oxidative stress is an important factor in male infertility, idiopathic infertility, as well as the disease condition of varicocele. And oxidative stress markers can be used as a diagnostic and prognostic tool for male infertility assessment and management. And within the ART setting, there is generation of oxidative stress, and several of the studies that I presented have led to adjustment of oxidative stress in the ART media, and this can improve the clinical outcomes of blastocyst development and pregnancy rates. So ORP, which is a new marker for oxidative stress and is easy to assess, is predictive of clinical outcomes in the ART setting. Well, I thank you for your attention, and this is our group in front of our center, and thanks very much.
And Dr. Sharma is now going to speak on evaluation of sperm DNA fragmentation, or also called as SDF, by lab developed test, or LDT. So he's going to take it over from here. Rakesh, you can begin.
Thank you, Dr. Agarwal. Good morning. This is going to be my second talk. The learning objectives are the tunnel assay by using a flow cytometer, understand the principle of flow cytometer, and more important, I'm going to highlight the terminology used in the measurement of sperm DNA fragmentation by flow cytometry, and also highlight some of the lab scenarios as well as the quality control in using sperm DNA fragmentation by flow cytometry. In the last 20 years, the 4 commonly published tests are illustrated in this figure. And as you can see, sperm DNA fragmentation testing by tunnel is the most widely published technique in the world. And sperm DNA fragmentation can be measured by 2 methods, using the fluorescence microscopy, in which we measure 200 to 500 sperm. This method is subjective, time-consuming, and many times inaccurate. The other method that is used is the flow cytometry. And in this method, 10,000 sperm are scored. It is objective, rapid, robust, and a very sensitive technique. And therefore, today I'm going to talk about the measurement of sperm DNA fragmentation using the flow cytometry method. The principle of flow cytometry is that the tunnel method measures both the single as well as the double strand DNA breaks. It's a direct method which tags with the enzyme binding to the sites which are broken or which have the 3-hydroxyl damage, and these can be measured using the flow cytometry. On the right is the figure giving a short protocol which has been discussed and shared with all of you. Here I would like to spend some time to highlight the principle of flow cytometry that we use in this benchtop flow cytometer, which is called the Accuray C6, and this is available through Becton and Dickinson. The basic principle of the flow cytometer is that the sperm are passing through a sheath fluid into single cells, and these are then excited using a blue argon laser, and the light that is reflected allows us to measure 2 important components, which is the forward scatter, which measures the size of the cell, the side scatter, which measures the granularity of the cell, and then based on the stain used for the tunnel, the absorption is by the green filter, which is the FL1 detector, and the propydium iodide, which is measured in the second filter, which is the FL2 filter. And here it shows the FL1, which is used to measure the green dye, which is FITC in this case, and the FL2 measures the red absorption which is coming out from the propydium iodide used for staining the nucleus. The signal is then amplified and then converted into a light signal, which then can be visualized on the computer. The difference between the benchtop C6 flow cytometer and the conventional flow cytometers is that this flow cytometer uses a peristaltic pump instead of the voltage detectors which are seen in conventional flow cytometers. This figure illustrates the kits, the reagents that are used, as well as the positive and the negative controls that come with the kit used for measuring the DNA fragmentation. Here I would like to spend some time in familiarizing you with some of the important terminology that is used when measuring the sperm DNA fragmentation using the flow cytometer. So this space that you see on the computer when measuring the DNA fragmentation is called the workspace. This allows us to measure each sample, and we provide the information over here. This is the plot, and on the screen you can create your own plots, 1, 2, 3, 4, 5 plots. And then we have the gating, which is the area that we allow for the cells to be measured. Anything outside this gate will not be recorded and measured by the instrument. This is also called a template. We create a template both for the kit as well as the test samples, and all cells will be measured within the template. The workspace is this area, and this area will allow us to not only record the information about the sample, but it also has the settings that we use. And the settings over here are that we run the sample measuring 10,000 events. We measure the P3 in P1, and what this means is that we are only capturing the cells which have complete nucleus, and we're excluding the cells which have apoptotic bodies that give us erroneous results. The fluidic speed is set to slow speed, and the threshold is set as 80,000. We also have the last plots showing the quadrant, and these are the 4 quadrants, the lower quadrants and the upper quadrants, and the extent of DNA damage is indicated by the upper right quadrant. In the flow cytometer, which is BDC6 Accurate, we make use of the virtual gate alignment, and in this case, a standard sample with a DNA fragmentation of less than 17% is used as the reference sample or the standard sample, and all test samples are then aligned with the standard sample to give us the final reading. And this is called the alignment strategy, and it is very important when running the samples for DNA fragmentation. So having understood these terminologies, I will take you to the next step. And this slide shows the most important step, which is the sperm preparation. For measuring tunnel using this method, we require a minimum of 2.5 million sperm. We also include a negative control, which is the samples in which we do not add the TDT, so there are no cells which are stained for DNA damage. We also include a positive sperm sample, and in this case, the positive sample is created by using hydrogen peroxide and incubating the sample for 1 hour at 50 degrees Celsius. The samples are permeabilized in 3.7% of paraformaldehyde and fixed before they are analyzed. Quality control is the first and the most important step before we even run the test samples. And this is important because the test measurement is an elaborate process, and it is important to know that the setting of the instrument as well as the reagents and the staining is optimized, and the quality control for the C6 is accomplished using the 8 peak validation beads. And essentially these beads provide us the 8 different peaks which can be measured for the 3 detectors, detector FL1, FL2, FL3. And the first plot shows the area where the events are tracked. We measure only the singlets and we exclude the doublets or the cells which are sticking together. The second plot will show the 8 peaks that are generated for each of this detector FL1, FL2, and FL3. And we measure the peak of the brightest fluorescence intensity, which is shown over here, and also check the coefficient of variation. The coefficient of variation should be less than 5% for the quality control to pass this criteria. Here I would like to show a quick video for the quality control. We take 500 cc of water and we add 4 drops of the quality control bead. Here is the gate, and we highlight the well in which the measurement is taken. We place the tube with the beads and we set the gate setting to 50,000 events. And here we are measuring the cells or the events, and these should be more than 70%. And here you can see the beads that are showing the 8 peaks, and we measure the fluorescence intensity of the brightest peak, and in this case it is 410,000, and this is showing the coefficient of variation of 1.49, which indicates that this quality control has passed the test, and now it is good to test the samples for DNA fragmentation. We make use of the kit control, and kit control contains the cells which are different than the sperm cells, and therefore the gating strategy will be different for the kit control compared to the sperm samples. And this plot shows the measurement of the negative and the positive kit control, and the quadrant for the negative kit control will give the value of 1.1%, which is negative for sperm DNA fragmentation because this is a negative control. Similarly, we will see the damage of 45.2% in the positive kit control, indicating that the 2 cells or the 2 samples are providing very distinct DNA fragmentation, and now we can measure the DNA for the test samples. For the test samples, again, the template creation of the template is very important. And once this template has been established, we always use the same template every time that the sample is run. The settings are set to 10,000 events. In P3 in P1, which as I explained earlier, we measure only those cells which are stained for the nucleus. The fluidic speed is set at slow, and we set the threshold to 80,000 events. And the measurement is done in the collect tab. It's very important to make sure that we are performing the test in the correct settings. So the measurement or the samples are run in the collect tab, and this workspace illustrates each well representing a single sample that is being tested. And these are the plots where the events will be recorded. We create this gate to exclude the debris from here. And the second gate indicates that all the cells in this gate are the ones which are having proper sperm nucleus, which are stained and are being measured. And the quadrant will actually give us the extent of DNA damage. In the collect tab, we also make use of the standard semen sample, as I explained, which has negative sperm DNA fragmentation, and each sample is then aligned using the virtual gate setting. And once this is done, all samples will be aligned to the standard sample, and the measurement will be done in the Analyze tab, where the 3 detectors are represented and will provide us the final value of the sperm DNA fragmentation for that given sample. And to illustrate this, I will show you a quick video clip. So here we see that the workspace is the unanalyzed. So here we have run the samples, but we have not analyzed the samples. And we are checking the distribution of the events in the gates. And here we make use of the standard sample, as I mentioned, and we will use the virtual gate setting to make sure that all test samples are aligned with the standard sample. And this is how the alignment is done. This is the standard sample. We place the marker right in the center of this peak, and then we use this to measure the alignment for the test sample. We pick out one test sample and we align the blue line over here to match that with the standard sample. So this is called the alignment using the virtual gate setting. And once the alignment is done, we will see an asterisk or a star, which indicates that the test sample has been aligned with the standard sample. This is very important because if the alignment is not done, then the distribution of sperm DNA fragmentation will be incorrect. Once this is done, we go to the Analyze tab, and in the Analyze tab, we plot the 3 detectors, and these 3 detectors are then shown over here, and we check the test that we are measuring, and this will provide us the events in plot 1, plot 2, and plot 3. And the plot 3 is the one where the sample has been analyzed, and the upper right-hand quadrant will indicate the extent of DNA fragmentation, which in this case is 18.6%. Each sample will be shown in the Analyze tab, and the last quadrant will indicate the extent of DNA damage. For example, the second sample has a DNA damage of 0.6%. So this is how the sample is analyzed and final results are documented. There are many factors which can affect the results. Number one is the accessibility of the stain to the DNA. Number two is the sperm preparation. Any errors or incorrect preparation will lead to erroneous results. The presence of dead cells can also impact the DNA fragmentation, the number of cells examined, as well as the inter and the intra-observer variation, and the inter and the intra-assay variation must be established within each lab. The figure on the right shows the receiver operating characteristic curves, which allow us to establish the cutoff. In this case, it is 16.8%, as well as the sensitivity and specificity of the test. And the figure on the right shows the distribution of the DNA fragmentation in the controls as well as the patients. And as you can see from here, all controls have a value less than 16.8%, whereas the patients show DNA fragmentation ranging from 16.8% to as high as 40 to 60%. This is how the Turner report is generated. We provide the information for the patient, the lab where the test was conducted, patient's information, and the test that was used, in this case, the flow cytometry using the C6 analyzer, and also the reference values for the normal as well as abnormal sperm DNA fragmentation, the display of the positive sample or the actual values of the sperm DNA fragmentation, and the results are interpreted for the clinician. Instrument quality control is extremely important in the measurement of sperm DNA fragmentation, and the quality control can be the different fluidics that are used, and these are the reagents used for the decontamination, cleaning, extended flow, as well as the bacteriostatic solution. As I mentioned, it's a peristaltic pump, so we have to be very careful that the fluidics is correct, the capillaries are not clogged, and these filters are changed at a regular frequency. Many times, the clogging can occur in which it will slow down the measurement of the events. And to troubleshoot, we place these fluidics in warm water, and then we flush the cycle, and this will clear the SIP and the measurement can be done. When we perform the sperm DNA fragmentation using the flow cytometry, there are many situations which can occur, and each of these situations I have illustrated as scenarios and also identified how we troubleshoot in each case. Scenario number one is that the quality control did not perform as expected in both the C6 or the C6 flow cytometer. The solution in this case is it's extremely important that we know the correct beads that are being used for the flow cytometer. If we are using the C6 flow cytometer, we must use the 8 peak beads, and the gating should include 75 to 80% of the events, and the coefficient of variation of the brightest peak should be less than 5%. If we are using C6 plus flow cytometer, then we do not use the 8 peak beads, we use the CST RUO beads, and the further adjustments can be accomplished using the zoom tool. Scenario number 2 is that a patient sample has a volume of 0.5 ml and a sperm concentration of 2 million per ml. How do we proceed? As I mentioned that we need 2.5 million sperm to perform this test because many sperm are lost with the washing steps. And in this case, the volume is only 0.5 ml, whereas we require 1.25 ml volume to have the desired 2.5 million sperm. So in this case, the tunnel test cannot be performed because of inadequate sperm concentration, and we will ask the patient to provide us another sample. Scenario number 3 is that the positive kit control shows a very low sperm DNA fragmentation of only 15% instead of the 30 to 40%, which is the desired percent of DNA damage. How do we overcome this situation? The solution here is that this is a test failure because the positive control did not give the desired percentage of DNA fragmentation. We make sure that we have used the correct template. We performed the staining steps correctly. The reagents were prepared correctly. And because it's a test failure, we should repeat this test again. Scenario number 4 is we have very few events which are recorded, or it is taking a long time to record 10,000 events. How do we proceed in this situation? The solution here is either the number of sperm in the sample are too low because they have been lost during the washing steps, or the sheath injection port or the SIP is clogged, and therefore the events are not being tracked by the laser. And in this situation, we will follow the back flush step and also clean and unclog the SIP and take the measurement again. So the take-home message for this test is that the standardized protocol, the quality control, the flow cytometer settings are extremely important. The test should be validated before it can be used as a diagnostic test. Based on our experience, we have created the templates, we have established the cutoff, and in our setting, a cutoff of 17% is what we use to determine the patient samples which have cutoff below this, or they are positive for sperm DNA fragmentation if they have a cutoff higher than 17%. And ideally, each lab should develop their own cutoff by testing a cohort of patients and proven fertile men. And this test, when used with flow cytometry as explained, is a very reproducible, robust test for measuring sperm DNA fragmentation using flow cytometry as a tool. So in the end, I would like to thank you for your attention. And this is one of our group picture and another view of the Cleveland Clinic. Thank you very much.
Now we want to move to our next talk, and the next speaker is Dr. Renata Finale. Renata is currently, she joined about 3 months ago as a researcher in our center at Cleveland Clinic. She has a doctorate in biotechnology of Reproduction from University of Rome in Italy. And she has received several scholarships and has published a large number of articles and papers in the field of male infertility and human reproduction. She is going to, again, just like Dr. Gupta, who put together the lab results of oxidative stress into clinical context. Renata is going in her presentation talk about the clinical importance of sperm DNA fragmentation, not only just the tunnel, but all different tests in human reproduction or assisted reproduction. So Renata, please go ahead.
Thank you very much, Dr. Agarwal, for your introduction. Good morning, good afternoon, good evening, everybody. So today, these are the learning objectives of my talk. Today, I'm going to discuss the sperm DNA fragmentation as a marker of male infertility and its association with natural and assisted reproductive outcomes. Particularly, I will discuss the association of this marker with miscarriage and pregnancy failure, and also I will present you the clinical practice guidelines which have been published by our group for sperm DNA fragmentation testing in male infertility. But let's start saying that sperm DNA fragmentation is not the only type of DNA damage which can occur in sperm. Sperm DNA can occur in mitochondria, in telomere. We can observe microdeletion of the Y chromosome as well as epigenetic abnormalities. However, in this talk, I will focus only on sperm DNA fragmentation, as it is the most widely studied types of DNA damage in sperm. And also, there are several studies investigating the association of this type of damage in sperm and clinical reproductive outcomes. So let's move. This is on the left side, as you can see, a picture of DNA. Sperm DNA fragmentation can occur at just one or both strands of DNA, resulting in single and double strand breaks. Here in this picture, I illustrate three different mechanisms which can result in sperm DNA fragmentation. For example, defective chromatin condensation, abortive apoptotic process, as well as the oxidative stress. All these three processes can lead to fragmentation at single or double strand breaks. This is particularly relevant in clinics because sperm DNA can be repaired until the third week of spermatogenesis, while there is no repair mechanism during the transit in the epididymis or post-ejaculation. Here on the right side, I report a table from an in vitro studies. In this study, as you can see, there are increased rate of sperm DNA fragmentation, and in the table it is reported its association with reproductive outcomes such as penetration rate, fertilization rate, or blastocyst development. As you can see, increased rates of sperm DNA fragmentation does not affect penetration rate or fertilization rate, while blastocyst development is significantly affected when sperm DNA fragmentation is higher than 88%. This is explained by the fact that the oocyte has limited ability to repair sperm DNA fragmentation, and the damage can be repaired only when it is less than 8%, as it is shown in this in vitro study. We should also consider that in the last years, the oocyte quality is declining worldwide, and this results in a declining capability of the oocyte to repair the sperm DNA damage. In general, high levels of sperm DNA fragmentation have been observed to be higher in infertile population, but however, a weak correlation has been reported between this parameter and the conventional semen parameters, which can be analyzed through the standard semen analysis, such as concentration, morphology, or motility. Here in this table, I reported the results from just 3 studies, but there is a huge literature showing this poor correlation. And this is explained by the fact that SDF is an advanced molecular marker of sperm quality. It is a molecular marker, and it is important, it is independent of conventional semen parameter. This is why these parameters should be analyzed along with the standard seminal analysis. But let's move on natural pregnancy, the impact of sperm DNA fragmentation on natural pregnancy. In this slide, I reported the results of a meta-analysis published in 2011 by Zini. In this meta-analysis, the 3 studies were included for more than 600 couples, as you can see in the table here on the right side. And this meta-analysis showed that high levels of sperm DNA fragmentation were associated with the failure to achieve natural pregnancy. However, we should not forget that this meta-analysis includes only 3 studies, a very small number, and also the confidence interval per single study is very large, suggesting a poor precision of the sperm DNA fragmentation in predicting natural pregnancy. Here, in this slide, I report the results from another meta-analysis published in 2019, very recently, and including a large number of studies, 13, for more than 500 men with a history of recurrent pregnancy loss. Here on the right side, you can see the forest plot from this meta-analysis. And this forest plot shows that couples in all the studies included, couples with a history of idiopathic recurrent pregnancy loss have higher levels of SDF than fertile couples. And here I'm not reporting the second forest plot, but the similar results were also shared by another meta-analysis published in the same year. Moving to the intrauterine insemination, a meta-analysis published 2 years ago, including almost 3,000 IUI cycles, reported an association of high and negative association of high levels of SDF and the lower pregnancy and delivery rate when IUI was performed. Here on the right side, you can see the forest plot illustrating the association of SDF and pregnancy rate, while on the right side, the forest plot illustrating the association of SDF and delivery rate. When IVF, in vitro fertilization, was performed, 4 different meta-analyses reported an association of high levels of SDF and lower pregnancy rates. In this table, I summarized the main findings from these 4 meta-analyses, such as also I reported the number of studies included, the number of cycles analyzed, and the main findings. And all 4 agree in reporting this negative association with the pregnancy rate when in vitro fertilization was performed. Also, when IVF was performed, a miscarriage rate was reported to be associated with high levels of SDF by 3 different meta-analyses. Here at the bottom, you can find the references, while on the left side, I reported just as an illustration the forest plot by Zhao et al. Published in 2014. Regarding the live birth rate was reported to be significantly lower in men with high levels of sperm DNA fragmentation after IVF. When ICSI was performed, miscarriage rate was also reported to be significantly increased when sperm DNA fragmentation was equal or higher than 30%. Also, the neutral comet assay was was suggested to be a good predictive technique for recurrent miscarriage. As you can see here in the ROC analysis, it showed high sensitivity and high specificity for predicting this outcome, miscarriage rate. Further, live birth rate, the association of live birth rate and SDF was analyzed in a meta-analysis published in 2015 and including 5 studies for more than 400 cycles. And this meta-analysis reported that men with low sperm DNA fragmentation had significantly higher live birth rate. However, we should not forget that all the studies that I've studied so far are not free of limitations. They are heterogeneous in terms of the studies included. They consider different threshold levels for defining high and low sperm DNA fragmentation. Sample sizes are always relatively small. An important consideration is that sperm DNA fragmentation is always measured in semen before the initiation of cycles. This means that the rate of SDF, which is measured, may not really reflect the status of sperm using for insemination. And also, in all these studies, bias and confounding factors are never taken into consideration when the results are analyzed. So let's summarize the current findings so far, described so far. In general, high levels of SDF have been associated with decreased pregnancy rate after in vitro fertilization and with increased miscarriage rate after. This can be explained by the fact that in ICSI, the natural fertilization process is totally bypassed by the embryologist. Also, it is possible that high quality oocytes may counterbalance, may offset the negative impact of sperm DNA fragmentation on pregnancy. Also, we should remember that in ICSI, gametes are cultured for less time than in IVF. This means that they are exposed to oxidative stress and laboratory-induced damage for less time. And another consideration is that DNA damage in sperm may affect genes which are responsible for embryo development and implantation, resulting in an increased miscarriage rate. So now I want to present you the clinical practice guidelines that have been published in last year in 2020 by our group. These guidelines were the results of a collaborative effort from 24 international experts in male infertility, and they were published based on the evidence already published in the literature for SDF testing. In these guidelines, we identified 5 different conditions, 5 different clinical scenarios which may really benefit from SDF testing. And these include recurrent miscarriage, lifestyle risk factors, varicocele, recurrent IUI or IVF failure, as well as unexplained and idiopathic infertility. For all these conditions, different type of treatment can be considered if SDF is significantly higher. And these type of treatments are summarized here in the picture. They include recurrent ejaculation, antioxidant therapy, lifestyle modifications, ICSI and the assisted reproductive techniques, or varicocelectomy in case of varicocele. So these are my take-home message. Don't forget that sperm DNA fragmentation is a functional marker of semen quality, of sperm chromatin quality. It cannot be analyzed by conventional semen analysis. High levels of fragmentation are associated with a longer time to achieve a natural pregnancy, as well as higher miscarriage rate. Also, high levels were associated with decreased pregnancy rate after IUI and IVF, and with increased miscarriage rate following IVF and ICSI. Finally, we provide clinical practice guidelines for the identification of those conditions which can really benefit from SDF testing. So thank you very much. This is a picture from our group in front of the Andrology Center, and this is a picture from the Cleveland Clinic. Thank you very much for the attention.
We will have now the next speaker, or he's the final speaker of our program today, Dr. Neil Parikh. Neil is a staff in the Department of Urology at Cleveland Clinic. He has done a fellowship in a clinical fellowship in andrology and male infertility, and was trained here recently before he went on to the staff position about more than a year ago. He has a lot of good experience in all aspects of male infertility, as you can see. More importantly, Neil has been part of the lectures, the online training program that we are hosting for the last 4 months, and he has helped us put all the laboratory-related questions in terms of the clinical interpretation and what should be the clinical management of patients as a result of the test results that we see. So Neil is going to begin his presentation today. Neil today is his surgery day. Always Tuesday is his surgery day. So he has kindly recorded his presentation, a wonderful presentation based on the questions that we had provided to him, and you will really enjoy it. So I welcome Neil to begin his presentation, or we play his recorded presentation here now.
Clinical Assistant Professor in the Department of Urology at the Cleveland Clinic. By now, you should have just heard Dr. Finale discuss the implications of sperm DNA fragmentation on assisted reproductive techniques. I'm going to segue into the clinical interpretation of these advanced sperm tests in my clinical practice in male infertility. We'll discuss a series of clinical questions as we've done before. We'll start with question number 1. A patient presents with primary infertility. This is his following semen analysis: a somewhat low sperm concentration at 12 million per milliliter, normal motility at 50%, a morphology of 6%, and an elevated oxidative reductive potential at 6.5 millivolts. How will we manage this patient? Now, this is a fairly common patient presentation. This patient has oligozoospermia and elevated ORP. And as you have already learned in previous lectures, this is abnormal. Just as we do in any workup of a male infertile patient, we begin with a thorough history and physical examination, as well as obtaining a social history, which includes everything from smoking history, obesity, drug use, etc. You want to identify any possible causes of the abnormal semen parameters, as well as elevated ORP. This can include varicoceles, infection or inflammation, retrograde ejaculation, cancer, a history of undescended testicle, testicular surgeries, hormonal imbalances, genetic mutations, pelvic surgeries, or potentially medication or radiation therapy. Now, up to 50% of patients, we do not quite know the etiology of male infertility. These patients are subsequently classified as having idiopathic male infertility. This is defined as the presence of altered semen characteristics without an identifiable cause and the absence of female factor infertility. However, and as we can see in this clinical situation, 30 to 80% of infertile men may also have concomitant elevated ROS levels. Elevated ROS levels are a potentially treatable condition, and this is discussed in our paper published last year called Male Oxidative Stress Infertility, the Proposed Terminology and Clinical Practice Guidelines for the Management of Idiopathic Male Infertility. So it is important, and I cannot stress enough, that male reproductive potential cannot be adequately assessed if seminal oxidative stress is overlooked. Unfortunately, there is no consensus on how to adequately measure oxidative stress or a diagnostic terminology until we published this paper on male oxidative stress infertility, or MOSI. So in this situation, this patient has abnormal semen parameters, but no identifiable cause of the abnormal semen parameters, and in addition has elevated oxidative stress, and which is indicated by his elevated ORP. So we start here with a basic semen analysis, followed by evaluation of oxidative stress through ORP. And if elevated, we want to begin by ruling out any clinical cause of the elevated OS, whether this is an infection, lifestyle issues such as smoking and obesity, or a clinical condition such as a varicocele. However, in a patient that does not have any reversible causes, but also has elevated oxidative stress, these patients are determined to have male oxidative stress infertility, or MOSI. Unfortunately, there are no evidence-based treatment guidelines available for the treatment of idiopathic male infertility. But we believe by identifying these men with MOSI, we can identify which group or which subset of patients would benefit from antioxidant therapy. So we know there are different types of empirical medical therapy, as I've discussed in my previous lectures, both hormonal and non-hormonal. Hormonal medication can be used off-label, which means not FDA approved in the United States, for the treatment of male infertility. However, the data on its efficacy is lacking. We do know many men are prescribed antioxidants regardless of their oxidative stress status. So many of these men who may not even require antioxidants may actually be being harmed by the overuse or overprescription of antioxidants, because we know that reductive stress, which is the opposite of oxidative stress, can have a negative impact of semen parameters. So we believe it is very important to identify these men specifically with elevated oxidative stress, and those men should be the ones targeted with antioxidant supplementation. We typically recommend a course of 3 months, and this is followed by retesting of oxidative stress with ORP after those 3 months. Clinical question number 2. A patient presents with primary infertility. He has an asymptomatic grade 2 left varicocele, and a semen analysis at an outside facility shows the following: normal semen volume of 2.5 mLs, a sperm concentration of 22 million per milliliter, a motility of 40%, and a morphology of 6%. How will you manage this patient? So in essence, this patient has normal semen parameters and a grade 2 left varicocele. If you remember, we grade varicoceles grade 1 through 3, and we typically only recommend fixing grade 2 and grade 3. But before we discuss any options, we want to complete the male fertility evaluation, assess the female factor, ensure that the couple is performing intercourse appropriately, timing it, not using any spermatotoxic lubricants, etc. If this patient were in my office, he has only had one prior semen analysis, which was normal, and an asymptomatic varicocele, I would recommend a repeat semen analysis and offer him advanced sperm functional testing. As you know, that the guidelines recommend 2 semen analyses spaced by approximately 2 to 4 weeks apart. And in this situation, the advanced sperm functional testing can give us more information before we decide to fix a varicocele. So here we have our treatment algorithm in this particular patient. His repeat semen analysis is here and shows a normal volume, a normal concentration, a normal motility at 42%, as well as morphology at 6%, but his sperm DNA fragmentation, measured via tunnel, is elevated at 26%. So after the evaluation of the infertile couple, we have identified a varicocele. Now, varicocelectomy is a treatment option in this patient with elevated SDF but normal semen parameters. The American Urological Association and the American Society of Reproductive Medicine also believes that varicocele treatment should be offered to the male partner of a couple attempting to conceive when all of the following is present: a palpable varicocele, which is considered a grade 2 or grade 3. A couple has documented infertility. The female has normal fertility or potentially correctable infertility, and one or more abnormal semen parameters or sperm function test results. So in this situation, while his semen parameters are normal, he does have elevated SDF. While the association between SDF and high-grade varicocele is much stronger, patients with low-grade varicocele also have shown an improvement in natural pregnancy rate that is similar to those with high-grade varicocele after surgery. Moving on to question number 3. We have a 28-year-old gentleman who presents with primary infertility for the last 3 years. There is no identifiable female factor. However, the couple has undergone 3 intrauterine insemination cycles without success. Here is his semen analysis. He has a low semen concentration at 9.8 million per milliliter, a reduced motility at 28%, elevated ROS levels, an elevated ORP, and an elevated sperm DNA fragmentation by tunnel at 36%. How will he manage this couple? So a number of societies recommend an endocrinological evaluation in a patient with sperm concentration less than 10 million, or with men with impaired sexual function, or if you suspect an endocrinopathy. So in this patient, the minimum lab studies I would recommend is a testosterone and FSH. However, I also routinely include LH, estradiol, prolactin, their thyroid function, and hemoglobin A1C if there is a concern for diabetes. In this particular case, this patient has no lifestyle risk factors to explain his poor semen analysis. He is a non-smoker, he's not obese, etc. His hormone profile was normal, so ultimately he is determined to have oligoacenozoospermia with elevated reactive oxygen species, ORP, and SDF. Now they have failed IUI 3 times, and we would consider him to have the diagnosis of male oxidative stress infertility. Again, that is a patient with abnormal semen parameters without an identifiable cause and elevated SDF or oxidative stress, as we see in this case. And as I showed you on the previous algorithm, we would recommend treatment with 3 months of antioxidants and repeat the semen analysis with advanced sperm testing at that point. Now, unfortunately, in this patient's case, he really had no significant improvement in his sperm parameters after 3 months of antioxidant therapy. His oxidative stress levels also remained elevated. Now, it is very common in the United States that IVF or ICSI not be covered by insurance and is a significant out-of-pocket cost for many couples. I know there are many listeners across the world, and it may differ from country to country, and in fact it differs from state to state within the United States. In Ohio, it is very rare that we can get adequate coverage for fertility treatment. So this is not an uncommon scenario for us. This couple, while they cannot afford IVF ICSI, would like to potentially try in utero insemination again, which is significantly more affordable here than IVF is. I would counsel them on the fact that there is a negative impact on prolonged ejaculatory abstinence on sperm DNA fragmentation. We have seen that short-term recurrent ejaculation is a simple, non-invasive option to improve sperm DNA fragmentation. In addition to higher pregnancy rates in ICSI, recurrent ejaculation has been associated with significantly lower sperm DNA fragmentation. So for this patient, I typically recommend a regimen where they ejaculate daily for 1 week and then abstain for 2 days before repeating in utero insemination. Now, this recurrent ejaculation, in theory, should reduce his sperm DNA fragmentation, and with the addition of antioxidants, may provide some benefit on repeating IUI. So here on the algorithm, we can see that they have had recurrent IUI failure. However, ICSI is not feasible for them, whether it is for financial constraints or religious reasons in other scenarios. However, recurrent ejaculation, as circled here in red, is a recommended treatment approach to lower sperm DNA fragmentation. Moving on to our final clinical scenario. We have a couple with no identifiable female factor who is experiencing recurrent pregnancy loss after ICSI. The male partner has shown normal semen parameters, however, has an elevated SDF by tunnel of 60%. How will you manage this couple? So we know that the use of testicular sperm for ICSI is thought or theorized to bypass post-testicular chromatin damage caused by oxidative stress during sperm transit through the epididymis. And that is the reason some may offer testicular sperm for ICSI. However, before we elect to or recommend testicular sperm extraction in a non-azospermic patient, as in this situation, we have to know that intratesticular alterations in chromatin remodeling can also coexist, resulting in testicular retrieval of sperm with fragmented DNA. So before we were to offer testi-ICSI in a patient like this, first-line therapy should be adequate clinical management of their SDF. So once again, identifying any reversible cause of their sperm DNA fragmentation and potentially treating with antioxidants if possible. Now, there are a number of articles published on the use of testi-ICSI in non-azospermic men with high SDF. However, the majority of these studies are very small cohorts or case series without adequate control groups, and they do not report on live birth rates. Further complicating the situation is that SDF testing has not been validated for testicular sperm, making interpretation of testicular sperm DNA fragmentation levels difficult. And while some studies report a positive impact on assisted reproductive technique outcomes using testicular sperm, the quality of the evidence is very weak. We also have to realize that we will be putting the patient typically under anesthesia for a surgical procedure that has potential risks. These risks include anesthesia, bleeding from a hematoma, infection. While these are all risks of a testicular sperm extraction procedure, proponents of testi-ICSI would argue that it is much less invasive than repeat oocyte retrieval in women. As of now, there is no consensus on the use of testicular-derived sperm for ICSI. So in this clinical situation, where the patient has normal semen parameters but elevated SDF, we would begin with first-line treatment, which is either antioxidants or identifying or counseling on reversible risk factors before we offer testi-ICSI. So we have gone over several clinical scenarios that I see on a routine basis in our male infertility clinic. I hope this has been beneficial for all of the listeners. And overall, the take-home messages of the lecture today include the fact that sperm DNA integrity is crucial for fertilization and the development of healthy offspring. And there are a number of scenarios which can lead to this, which include men with idiopathic infertility or unexplained male infertility, recurrent pregnancy loss, clinical varicocele, IUI or IVF failure, and modifiable risk factors should all undergo sperm DNA fragment testing. We do have a few treatment options that are available to us at this time. This includes recurrent ejaculation, antioxidant therapy, lifestyle modification, and varicocelectomy are all proven treatments in reducing SDF. We know that reducing SDF seems to translate into better reproductive outcomes, and we also know that there are significantly lower levels of SDF in the testicle compared to ejaculated sperm. However, we need more well-designed studies in order to make a definitive conclusion on the use of testicular-derived sperm in ICSI in non-azospermic male with ICSI failure. Thank you for listening. If there are any questions, do not hesitate to reach out. My email address is Parikh, PAREKHN3 @ccf.org. Thank you.
So now my goal is to put your finishing touches and then move to the next part of the program, which is the multiple choice test. So let me begin to summarize the entire series of talks that we have given. We had 6 lectures which were given today, starting with Dr. Ralph Henkel, who talked about the advanced test, which provide useful and important information on sperm fertilizing capacity. Then there is an imbalance between oxidant and antioxidant that can result in oxidative stress, which is a recognized cause of male infertility, leading to a variety of damage to the cell components. Then we listened to the talk from Dr. Sharma on sperm chromatin structure test, tunnel, SCD, COMET, and many other tests which can be used for testing the sperm DNA fragmentation in a laboratory setting. There is no consensus as to what test is really the best, although we know that the tunnel test is widely reported, widely published. It can be done both by fluorescence microscopy and flow cytometry. Flow cytometry is considered much more robust and reproducible because you can count 10,000 cells. So it is definitely provides a higher degree of sensitivity. ORP is a new marker, a reliable marker of oxidative stress status. Is a new test. It's very simple, very easy, and it can differentiate between fertile and infertile patients with normal and abnormal semen characteristic with great deal of accuracy, and the results are reliable in settings in different parts of the world or different populations. Furthermore, high levels of oxidation reduction potential indicates MOSI or male oxidative stress infertility as described by Dr. Neil Parikh, and this is treatable. And we know now ORP level is predictive of assisted reproductive outcome as shown by Dr. Gupta. Adjustment of oxidative stress in assisted reproduction media can improve blastocyst development and pregnancy rate as discussed by Dr. Gupta from the work of our colleague, Dr. Israel Maldonado from Mexico City. And that high SDF levels or high sperm DNA fragmentation is associated with poor reproductive outcome in natural pregnancy and after IUI and IVF, as shown by our young investigator, Dr. Renata Finale. High levels of SDF is correlated with low live birth rate and high miscarriage, as reported by Dr. Renata from a variety of meta-analysis and clinical guidelines which have been published. And furthermore, the sperm DNA fragmentation testing is most beneficial in patients with unexplained and idiopathic infertility, in recurrent pregnancy loss, varicocele, patients who are opting for assisted reproduction, and in those with lifestyle or environmental risk factors. So these are the type of patient which will benefit maximally by sperm DNA fragmentation testing. Very important thing which I wanted to stress here, whatever test that you do, it is important that you understand that most of the tests which are in the area of reproductive field, whether in IVF or andrology, or specifically most of the andrology tests which are bioassays are not standardized. So it is important that those tests, the laboratories take the onus to to develop the protocol. They identify the quality control scenario, have positive and negative standard to establish the test accuracy from the laboratory. And then they need to embark on a clinical validation by testing the patient's samples, both negative and positive, and also fertile men, and develop a ROC analysis or a receiver operating characteristics to develop a cutoff value above which the test results can be considered as abnormal and below which it would be normal, to develop the sensitivity, specificity, negative predictive value, and positive predictive value. And furthermore, they need to test for intra and inter-observer variability, assay variability, intra and inter-lab variability, and establish acceptable criteria for intra and inter-lab coefficient of variation. So these are very, very important aspect of using a test by simply taking any number of tests that are being reported or conducted by various clinics or centers, and then using them. I think you will find that those results are not going to be reliable because they have not been standardized in your setting, among your own patient, among your own instrument, among your own personnel. So the test results can be highly, highly inaccurate, whether you are doing the SCD test, whether you are doing the tunnel test, the SCSA test. These are extremely precise tests, and they have to be done under a very robust protocol and settings, and standardization of the test is a must. Without that, the test results are going to be completely inaccurate. Now, I just wanted to talk about our next online test, online module that we are going to be starting in May. It's called Module 5A, which will cover both IVF and ICSI. And the learning objectives of this particular test, online module is on Tuesday, May 11th. And we are going to cover all the essential equipment, supplies, and culture medium, protocol for oocyte retrieval, oocyte denudation, embryo transfer, oocyte characteristic, embryo assessment, important parameter for embryo culture, monitoring, and quality control. So I think this will be a wonderful program that we are planning for many, many months now already. And this is the detail which we are going to now share with all of you on LinkedIn, as well as when you go to register for the next course, you will see the entire program. Our speakers are going to be Dr. Christina, who is a senior clinical embryologist in one of the very important IVF program in Athens, Greece. She's certified as a clinical embryologist from ASHRAE and has over a long experience in the field. She has studied at National University of Athens and trained as a clinical embryologist in UK and also at Cleveland Clinic. She has been providing IVF training at the American Center for Reproductive Medicine for almost 10 years as the chief training faculty. Our second course faculty is Dr. Israel Maldonado. Israel is the owner and CEO of the IVF clinic Cytometry with branches in many parts of Mexico. His main headquarters is in Mexico City. He is trained as a clinical embryologist in Valencia in EV program, one of the most prestigious IVF program in Spain, and has received additional training at the Keto Ladies Clinic in Japan. Israel has been recognized recently as the Embryologist of the Month by the American College of Embryologists, and he serves as a visiting faculty in ART training program of our center, and we have a long relationship. Both of them are superb embryologists and trainers, and they are extremely clever and smart in the field of IVF. So this is our ongoing program. As you will see, after the IVF ICSI, we go into the month of July, where we are going to cover oocyte and embryo handling. In September, we have embryo biopsy. In November, we have PGD. Then come January of next year, we have embryo cryopreservation and so on and so forth. So the entire training will continue until the end of November of next year. So I want to thank very quickly our course faculty, Professor Henkel, Dr. Gupta, Dr. Renata, Dr. Sharma, and Dr. Neil for their fabulous program, for enormous amount of effort and time they have spent. Our program coordinator, as you see on the right side, who have spent a tremendous amount of time on a daily basis putting this program together. This is our ACRM team, which we took these pictures this morning. We had to take our mask just briefly, and I'm just sitting here in my office, so I've taken the mask. We are still wearing, although we have been immunized. We have received our vaccination, but the policies requires that we continue to practice wearing the mask and social distancing. So this is the front of our andrology center, and this is where the patients come to get all their testing done, and we receive patients pretty much every day. So I want to also thank our colleagues here, Dr. Damayanti, and Dr. Christian Lysigang for creating the online survey, but more importantly for supporting a variety of things for this program. Specifically, they went through our multiple choice questions multiple times to review and edit and clean the things so that they are completely clear. Our lab technologists and our colleagues from the IT department who helped with the program. I want to tell that putting this program is a huge exercise. We were just cataloging the amount of time that we have spent. We have roughly spent among about 15 or 18 of us who are part of the program, close to 300 hours over the last almost 2 months since we started preparing for this. And this is very important for all of you to know. So this type of program is a very high quality program. It takes enormous amount of resources and time, which is in addition to our regular duties, which we are all extremely busy as well. But more importantly, I wanted to tell you a similar program at the Cleveland Clinic is being offered at a cost of between $300 to $500 US. So this program comes completely free to all of you.